Yeah, good afternoon, and thank you for that introduction, and thanks to the chairpersons. For want of time, I will not take much of your time, with the introduction, but for saying that, my thanks to the organizers. So in the next 20 minutes or so, I would like to share some of our research with respect to the spices and their novel bioactive compounds, as well as their potential against diabetes and its complications, particularly complications of diabetes and obesity. So this is the aerial view of my institute. Well, again, I will not spend much time here because we all know that the magnitude of diabetes, but I would like to focus that, uncontrolled prolonged diabetes leads to various complications, which we call it as long-term complications or secondary complications, etc., etc. Because diabetes affects many organs of the system, such as eyes, kidney, heart, brain, etc., etc. So these complications are classified as non-vascular and vascular complications for simple reason that, wherever there is involvement of blood vessels, we call it as a vascular complications, otherwise non-vascular complications. In again vascular complications, we have macrovascular and microvascular complications. As I said, when a person is diabetes for a prolonged period, almost every organ of the body gets affected, mainly eyes, kidney, heart, feet, etc., etc. In this background, we have been working with a set of complications as listed here: retinopathy that affects the retina of the eye because of the diabetes, cataract that affects the eye lens, nephropathy, the kidney, and neuropathy, of course, neural involvement, mainly nerves. The reason why we are working here is that, control of glucose is very important for the prevention of complications. Particularly, how well you maintain your glucose levels by whatever the means. It could be anti-diabetic medication, it could be insulin, it could be physical activity, etc., etc. But practically, it is sometimes not always possible for various reasons. You may avoid sometime insulin injections, or you may be off the track of your drugs, or sometimes their efficacy may be a problem. On top of that, we have something like insulin resistance, metabolic syndrome, or obesity. They are all related terms, which typically do not result in a high glucose in the blood. But the undercurrent is already there because insulin resistance, metabolic syndrome, and obesity are very much associated with type 2 diabetes. As I mentioned, there is always a fluctuation in glucose levels. Therefore, in addition to anti-diabetic medication, we may require adjunctive therapies based on some molecular process, which I will share in my next slide. Now, what are those molecular process? How can we control these diabetic complications? That's what I am going to share. So these are the molecular pathways that are linked to the development of diabetic complications, particularly the uncontrolled glucose levels. We all know that the normal range of glucose that we get, 80 to 100 mg, is usually metabolized through these typical pathways that we have studied: glycolysis, TCA cycle, etc., etc. But when you have excess glucose, more than 100 mg or 110, whatever it is, in diabetic people it could be 200, 300, and 400. So when you have excess glucose, this glucose gets sent to many other pathways which are shown here. I will focus on only two of them. When you have excess glucose, this glucose gets converted to its alcohol form called sorbitol by an enzyme called Aldo reductase, or this sorbitol is also called as polyol. That's why this pathway is named as polyol pathway or sorbitol pathway. Under excess glucose conditions, you get accumulation of sorbitol in a set of tissues that I will show you, particularly which are not controlled by insulin, because your glucose uptake is mediated by insulin. But there are some cells in the body, whether renal cells or eye cells, retinal cells, their glucose intake is not regulated by insulin. Therefore, when you have excess glucose, can you hear me? Yeah. Okay. When you have excess glucose, so the glucose gets converted to its alcohol form. When you also have excess glucose, it's not only glucose, some of the glucose derivatives, whether it is glucose 6-phosphate, fructose, or fructose 6-phosphate, some of these glucose derivatives are also accumulated in the system. So these glucose and glucose derivatives bind to some long-lived proteins or slow turnover proteins, and these proteins are modified by these sugar derivatives, which we call it as advanced glycation end product. In simple terms, we can call it as AGE, or this pathway is called AGE pathway. So obviously these pathways, whether polyol pathway, AG pathway, I'm not talking about the exozyme pathway and protease kinase C pathway. These pathways have become targets for the diabetic complication, drug targets. When I say targets, drug targets. People are working how to control the shunting of the glucose through these pathways. I'll pick up the first pathway, as I mentioned, where glucose is converted to its alcohol form, that is sorbitol, by an enzyme called aldose reductase. So this enzyme and this pathway is very peculiar. Under normal conditions, or under normal physiological conditions, the significance of this pathway is really not known. And this enzyme does not work because the Km for this enzyme, glucose Km for this enzyme, is very high. Therefore, one has to really have high glucose for it to act on glucose. Anyway, in diabetic conditions, we have high glucose and it gets converted to sorbitol. Obviously, we want to stop this conversion so as to protect the cells from osmotic stress, or for that matter, the downstream effects of the sorbitol. There has been a lot of work on the inhibitors of this particular enzyme called aldose reductase, or ALR2. The human form of aldose reductase is called ALR2 because there are many aldose reductases. So these molecules are like, toleristat, fideristats, etc. are widely investigated, and they are very successful whether in an in vitro system like cell culture or in animal models, preclinical models. But for some reason or the other, by and large, these molecules are failure in clinical trials. So, and also there's a lack of efficiency and specificity. That's why there's a lot of adverse effects. There is a need to identify newer, more specific, more efficacious aldose reductase inhibitors. Similarly, the other mechanism that I was talking, when you have excess glucose or glucose derivatives, they react with the primary amine group of the proteins, and the first the product of this reaction is a skip base, and this skip base undergoes various transformations and then results in something called a highly modified protein called advanced glycation end product. And these modified proteins, which I keep on saying AG modified proteins, this modification leads to alteration in their structure and function if all these proteins have some function. So therefore, again, this has become a target for us in the sense that how can we prevent the modification of these proteins, or in other words, the interaction of the sugars with these proteins. There have been limited studies, with respect to some compounds such as aminoguanidine and PTPs, etc., etc. People have tried, but again, at a clinical stage, this story is not really very, very successful. So in this background, as naturally, like Professor Padmanabhan, or for that matter, anybody says in our context, we have a lot of treasure with respect to spices or other functional foods. So we decided to explore some of these spices and their bioactive components, whether any of them have got potential molecules in them to prevent the polyol pathway as well as to stop the conversion of the or modification of the proteins. So therefore, we have used a large number of sources, versional sources to screen, because we have developed a screening assay for these two mechanisms. So we have screened, and in some cases, we could isolate and characterize the effective and safe aldose reductase inhibitors and anti-glycating agents. When I say anti-glycating agents, the agents that prevent the modification of these AGs. So from spices and functional foods. For this talk, I am going to focus on spices, but since I am from National Institute of Nutrition, not only for spices, we screen a lot of functional foods and dietary sources. You may see what all the things that we have screened in the next slide. Before I show you the slide, how do we do that? For aldose reductase, basically we have cloned this human aldose reductase, and we express in bacteria. We have pure recombinant aldose reductase, which can be assayed in any test tube or in cuvette. But of course, I will show you animal studies later. So using this simple recombinant human aldose reductase assay in the lab, we either screen the extracts, or wherever there's a scope for us to isolate the molecules, we also test their efficacy to see the inhibition. For example, I'll show you the extract of the pepper or cumin, how it would screen against this assay and to find out what is the level of inhibition. And wherever we have a pure molecule like curcumin, as Professor Padmanabhan said, to see whether it inhibits aldose reductase. If so, what kind of mechanism it inhibits, and we can also derive the binding kinetics, etc., etc. But I will not go into the deeper details of these mechanism aspects, but I will only show you what all the bioactive compounds that we could figure out from the sources that we have screened. By using this kind of assays, like as I said, by now we have screened close to 200 different functional foods or spices or dietary sources. the arrows that I said are focusing towards the spices, but otherwise we have variety of sources that we have screened over the years. Almost 200 dietary sources are functional foods. So to begin with, either aqueous extracts or different solvent extracts, then figure out which extracts have given the best inhibition. Then you pick up that extract and go back to your activation, characterization of the active molecules. So we have done this with respect to a few sources, but for today's talk, I will share the studies with only two sources. One is that because it is very interesting that one of them happened to be the Amla or gooseberry that we call it as, or Amla Extract in scientific terms. Therefore, we wanted to see, because this extract has given a promising inhibition with respect to the enzyme that I mentioned. This is the inhibition curve. First, when you see the inhibition with Amla extract, naturally we all know that Amla means vitamin C comes into our mind, and everybody suspects. So also we suspected, is it possible that vitamin C could inhibit this enzyme? But you see the table here. Now all the way up to 5 millimolar vitamin C doesn't inhibit this enzyme. That means it is not vitamin C, something else in this extract that is inhibiting the enzyme that we are interested. So then we went back to the bioassay guided fractionation. I will not go into the details of this fractionation, that how we have done. And we picked up the active fractions from this bioassay guided fractionation. The active fractions we subjected to HPLC, wherein we could find the activity in 3 peaks. But of course, we picked up the major one, that is peak 1, where we had got the maximum activity, and use that to characterize by mass spectrometry. Of course, we have also characterized the other 2 peaks also, because they are also giving the inhibition. So we realized that this particular fraction happened to be a molecule called gallyl beta glucose. Very interesting, actually. The reaction what I wanted to stop is a conversion of glucose to sorbitol, and the molecule that we find in this source is a derivative of glucose. So that is nothing but a gallyl moiety sitting on the glucose. So by this mass spec, as well as by H1 NMR, C13 NMR, and even by elemental analysis, because this is the first time we are finding the gallyl glucose in Amla. So we wanted to make sure that by all means, it is gallyl glucose that is present in Amla. So once we are sure that it is a gallyl glucose, that is the one which is inhibiting the Aldo's reductase. Then next story, as Professor Padmanabhan says many a times, the in vitro assays can be non-specific, and it might be simply inhibiting the assay that I am using. So therefore, we wanted to check, is it possible that this works in vivo system? So what we did, we used this human Aldo's reductase ALR2 or AKR1 transgenic model. It has been cloned under a particular promoter and then put it in a mouse lens. The reason we choose mouse lens is that mouse lens expresses a very, very low amount of Aldo's reductase. We thought that's the best way to express this one. Anyway, I will not again go into that genomic aspects. Using this transgenic mouse, and which when you put it under high glucose, because you have Aldo's reductase, and this high glucose is got converted to the slot of sorbitol. You see the normal mouse, whereas this transgenic mice, which is expressing Aldo's reductase, you get a lot of sorbitol. Now, we have given our beta gallyl glucose to this one and see whether this sorbitol gets suppressed. Yes, you see a significant suppression of sorbitol. That means what we have identified and shown the activity in vitro also works in vivo. This is something interesting. But then we also wanted to see how is this better than the previous ones. As I said, there are a lot of previous molecules. The previous molecules happen to bind to one of the 2 sites of the active, 2 pockets of the active site, the small molecules. Whereas our molecule happened to be occupying the both pockets of the active site. Maybe that's why it is better. Otherwise, what happens in this case of small molecule, pocket gets closed. This is called closed inhibition, whereas this is open inhibition. So open inhibition is supposed to give the better efficacy and supposed to be more specific, because this kind of inhibition is similar with some other related variables. So this is why we have identified a novel molecule, and this also gave a lead for us to see if we can even modify this and get much better activity. So that's what we have done with the help of some of the colleagues at IIT Madras, where Professor Indrapal Singh has developed a lot of analogs, and these analogs seems to be much stable and much better. So right now we are working with various analogs, but obviously we found a novel molecule in Amlica, which is a specific inhibitor for Aldo's reductase. So from as said, as another spice that while at the same time we were working with the black pepper, and my colleagues. So, yeah, actually, this is a part of work was going on at the Indian Institute of Chemical Technology. My colleagues were working on pepper molecules, and they already fractionated quite a few molecules. Then we got curious to understand whether any of them have got the potential to inhibit this enzyme. And one of the molecule from this pepper happened to be the pipelatine, which inhibited Aldo's reductase. But we are not very happy with the IC50 value. Therefore, we wanted to make much more specific and more efficacious analogs. Of course, they synthesize almost 50, 60 different analogs using the clue that I showed earlier. How can and what molecule we can place on that and how they can occupy this active site. That way, from the 50, 60 molecules, we figured out that 2 or 3 happen to give almost 40-fold increased activity, starting from 160 micromolar to we could come down to 4 micromolar. So by this engineering. So these molecules we have patented because these are not natural molecules. They allowed us to patent, whereas natural molecule cannot be patented. The other pathway that I have been talking is the prevention of protein modifications. For that, again, we have a very, very simple screening assays in the lab. Here, what do? We take any protein, like you can take a BSA, you can take lysozyme, and incubate them with any of the sugars that I mentioned, glucose or fructose or glucose phosphate, and then remove the unbound sugar, and then assess the level of glycation by different methods, because it is not a straightforward enzyme assay that I showed in the previous essay, because it can give a spurious results. Therefore, we have used a complementary methods like 4 or 5 different methods, where we can see the fluorescence coming due to the modified protein, the protein oxidation due to the modification, even the quantification of the modified protein, and also we have developed specific antibodies, in-house developed antibodies to this modified proteins. By using all these methods, we have screened about 30, 40 dietary sources or spices, because it's a very laborious, unlike the previous Aldo's reductase assay, which is very simple. You can screen hundreds and hundreds, but it is very, very cumbersome. Therefore, we could screen only 30, 40, and I'm showing the best ones in this table. And out of them, I picked up cinnamon. Of course, we are also working with others, but I'll show you the data with the cinnamon. So we were curious what molecule in cinnamon is inhibiting these reactions. Again, I will not bother you with this fractionation aspect. Using all the 4 or 5 essays that I showed in the previous slide, whether AG fluorescence, cross-linking, or you can quantify the modified proteins by affinity chromatography, we could narrow down to a couple of molecules in cinnamon. One is procyanidin B2, another one is ellagic acid. These 2 molecules are the one that happen to be the molecules that prevent the reactions that I showed you, the sugars with the amino groups of the proteins. So as I said, we have, again, using the mass spectrometry and HPLC, we have really nailed down that it is a procyanidin B2, because in procyanidins, you have different procyanidins, A series and B series. In again, B, you have B1, B2, etc. It happened to be one particular B2 in this source that is inhibiting this particular protein modification. Now, so far, so good. What do I do with this one? So we want to relate it to the actually translational aspect. So as I was talking, my problem was diabetic complications. So we create diabetes in animals, and we allow the animals to live with the diabetes for a couple of months or 3 to 4 months, where they develop eye problem or kidney problem. Now we feed these animals, whether the original source or the isolated molecule, and see whether we can prevent these complications or we can even reverse if possible. Our first approach was to see whether we can prevent. Then we have also taken the therapeutic approach. So we have got all the technology, methodology, and tools to do that, whether it's a retinal dystrophy, whether a renal problem or a cataract problem. We have all the expertise with us. So by this way, not only the one that I told you, procyanidin B2 and gallal glucose from Amla, we have also figured out curcumin and rutin from other sources. We have all the studies, but what in a summary, I am trying to show that when you feed these molecules, whether the original source or the active molecule, because we can relate how much active molecule one has to feed based on the original source. That's how we have done these animal studies. And we could slow down the diabetic complications, whether it's a diabetic cataract or whether it's a diabetic retinopathy. In case of diabetic retinopathy, we see that, I'm showing a couple of markers here for want of time. In diabetic retinopathy, there's a marker called glial fibrillary acidic protein. Due to diabetes, it gets upregulated. and vascular endothelial growth factor. It is involved in many vascular problems, including diabetic retinopathy. Compared to control, you could see a lot of signal in diabetic retina. But when you give cinnamon or its active molecule procyanidin B2, almost it is as good as control retina. So also with respect to glial fibrillary acidic protein, you see a lot of signal in the diabetic retina, but it's not the case with procyanidin fed animals, which indicates that these molecules have got some potential to stop this vasculature in the retina due to diabetes. So also with nephropathy, where again similar molecules we could see, but instead of showing the same molecules, here I want to show you something interesting. That one is the CML, one of the modified proteins. We could see that which we could completely stop the conversion of the proteins to CML adductor. In other words, this is the one. CML means a particular form of modified protein. You see a lot of modified proteins in the kidney, but because of the ellagic acid, you see as good as the normal kidney profile. So I will not go into other markers. Basically, I picked up a lot of molecules with respect to renal failure. What happens in diabetes? Then how could we prevent these changes by these molecules? That's the some of the our message of this slide. At the end of it, what I want to convey to you is that we have screened functional foods or spices, and in some cases we have characterized novel models indicate the potential of these molecules in preventing and arresting the progression of diabetic complications. Therefore, ingredients of spices or functional foods could be explored for their utility against diabetic complications. And in some way or the other, these complications will also occur with obesity and metabolic syndrome. Therefore, this can also be used for those complications that are associated with obesity. And one major problem with these functional foods and their bioactive molecule is their bioavailability, like as Professor Padmanabhan said. Therefore, we are also using the same technology, the nano-emulsification of some of these isolated molecules, or for that matter, Professor Indrapal Singh is actually coming up with analogs which are better bioavailability and better stability. So that's where we are at this point of time. So I will stop here, and if there are any questions, I will try to answer. Otherwise, this is the funding source that we get from all the government agencies. And as I said, these are the collaborators for this particular work. Of course, we collaborate with many people, but this particular work that I shared with you, we collaborate with these groups. Thank you very much.